set of talks next for this final session. The first one we have is ketamine for the treatment of depression and Parkinson's disease from Sophie Holmes. Thank you, Sophie.
Can you hear me? Ish? No. Okay, I'll try, but shout if I'm mumbling, because I tend to. So thank you, Rupert, for the invitation. I'll be presenting on a clinical trial that Jerry Sanacora and I are running in Parkinson's disease, but we're halfway through, so I'll be presenting the rationale, going through the study design, and then some initial blinded results. So first I'm going to present some PET imaging work in depression and ketamine and then Parkinson's, which provides the mechanistic rationale for the trial. So one of the major neurobiological underpinnings of depression is thought to be a loss of synaptic connections. And evidence for this comes from preclinical work, much of which was led by Ron Juman at Yale, showing that chronically stressing out rats results in dendritic atrophy and a loss of synapses and associated depressive-like behavior. And he also conducted postmortem work showing lower number of synapses and synapse-related genes in MDD. And then indirect evidence comes from MRI studies showing structural and functional alterations converging on the corticolimbic circuitry that may be underpinned by a loss of synaptic connections. But more direct evidence in humans is lacking, in humans that are alive. And we can do this now with PET imaging and a radiotracer that binds to the synaptic vesicle protein 2A, SP2A, which is located on synapses, every synaptic vesicle, so acts as a marker of synaptic density. So we used this radiotracer and PET to show lower synaptic density across mood-related regions in people with depression, and a correlation between binding in the DLPFC and depression severity, so lower number of synapses in DLPFC associated with more severe depression. So this is one of the first in vivo evidence for a loss of synaptic connections potentially contributing to depression, which is a target for ketamine. So chronically stressing out rats results in synaptic loss. Ketamine reverses this synaptic loss and can reverse depressive-like behavior. And since this, a lot of preclinical work has shown that ketamine has synaptogenic and neuroplastic effects. But this has largely been limited to preclinical work. So we looked at this in humans in vivo using PET. So this was our study design. This was work with Irina Estelis. So we had 10 controls and 12 patients with MDD, and some had comorbid PTSD as well. We did baseline UCBJ, and then on the same day, they were infused a single dose, 0.5 mg per kg, and then the next day they came back 24 hours later for a post-treatment scan or post-infusion scan. We chose 24 hours because that's when we see peak clinical effects. So David Nutt showed this earlier, So in controls, we didn't see any increase in synapses after a single dose, 24 hours after a single dose of ketamine. Across DLPFC, anterior cingulate or hippocampus. And then in patients, we also overall did not see an effect, which was disappointing. But we then did a secondary analysis. So we stratified the patients according to their baseline synaptic density. So those that showed a deficit in their baseline synapses, we saw an increase in synaptic density 24 hours later. So that's in the red graph. You can see a % increase across ROIs in that subset. So it's a small sample, but what made us slightly more confident this is real is that the correlations make sense in terms of direction. So the % increase in synaptic density in the anterior cingulate was associated with antidepressant response. The % increase also was associated with derealization, and the antidepressant response was associated with derealization as well. We don't think that ketamine is, that the increase in synaptic density is related to the dissociative symptoms directly, but we think it could be evidence of target engagement. So the ketamine results in a glutamate surge, can cause dissociative symptoms. Then 24 hours later, synapses can regenerate, hopefully, maybe, and that's associated with the antidepressant response. Okay, so that's mechanistic rationale for Parkinson's disease study that we're doing. So Parkinson's is characterized by motor symptoms, but there are these non-motor symptoms that are very much part of the disease, and depression is one of the most significant. It affects half of people with Parkinson's, really impacts quality of life. One of the biggest Parkinson's outcome studies show that depression and anxiety are the top factors affecting overall health. It also impacts motor symptoms and can accelerate disease progression, so it's really important that we treat it effectively. But traditional antidepressants are generally not that effective in Parkinson's, likely because they're not targeting the underlying neurobiology. So there's a critical need to develop targeted treatments in Parkinson's. So we need to understand the mechanisms. So Parkinson's is diagnosed when the motor symptoms develop, but the disease process is thought to begin like 20 years before this, and non-motor symptoms such as constipation, REM sleep behavior disorder are apparent before motor symptoms develop. And depression often develops about 5 years before the onset of the motor symptoms, suggesting that it's related to the pathology of Parkinson's itself, which is a buildup of misfolded protein, alpha-synuclein, which can enter the brain, affects brainstem regions, and then is toxic to neurons in the substantia nigra that is responsible for the motor symptoms. So using UCBJ PET, this is just in Parkinson's, and controls, we showed, the red arrow is substantia nigra, and then you can see a much lower signal in Parkinson's disease. So this confirmed in vivo what we know from post-mortem work and about the pathology of Parkinson's. And importantly, we see a correlation as well. So lower synaptic density in the substantia nigra is associated with more severe motor symptoms. But it's thought that pathology outside of the substantia nigra is responsible for the non-motor symptoms, including depression. So our hypothesis is that synaptic dysfunction in brainstem nuclei and projecting to corticolimbic circuitry could be the substrate for depression in Parkinson's disease. And we have preliminary evidence, again using UCBJ, showing that in people with Parkinson's and depression, they have lower synaptic density in mood-related regions and a correlation between DLPFC, synaptic density, and depression severity across people with Parkinson's disease. So potentially different pathway leading to synaptic dysfunction in mood-related circuitry, but potentially a final common pathway that we can treat with ketamine. Michael J. Fox have funded a clinical trial that I'm running with Jerry Sanacora. And this is our study design. So we're randomizing 50 patients with Parkinson's disease and depression to receive 6 infusions twice per week of ketamine or placebo. Primary outcome is MADRS. We'll also look at anxiety, apathy, pain, and Parkinson's symptoms themselves as well. And we're doing brain imaging, so looking at synaptic density and fMRI network organization pre and post. Our hypothesis being that ketamine can increase synaptic density in mood-related regions, potentially other regions as well that could have symptomatic effects, and that this is going to impact the organization of the brain that we can measure with fMRI. So far we've randomized 25 people, and this is blinded data, but we're seeing quite a big response. So 68% response rate and 58% are in remission. And then this is with 1 month follow-up. Some people stay well, and some people, the effect starts to wane. So I think this is somewhere we can intervene to sustain the effects, so that the response and remission rates at 1 month. So the premise of ketamine's antidepressant effects, or one of them, is that it results in neuroplasticity and allows for more flexible and helpful ways of thinking. And while the brain is in a more plastic state, this is a window of opportunity to intervene psychologically. And this some work led by Sam Wilkinson and Jerry have shown that CBT can sustain the antidepressant effects of ketamine. So we have just got some internal Yale funding to add an extension to the trial. So after the infusions, patients will have 10 weekly sessions of CBT. And this is CBT that was specifically designed for use in Parkinson's disease. A psychologist that we work with at Rutgers has developed a manual specifically for Parkinson's because they have unique challenges. And then we'll follow people up. We'll keep the 1 month follow-up, but we've also added some later time points as well. And the idea is that we can try and link this back to the imaging. Do we see that people who show changes in synaptic connectivity and neuroplasticity, are they the people that will respond most to CBT? That's our hypothesis. So I've still got loads of time. Okay. So placebo response in Parkinson's. People with Parkinson's are thought to be more susceptible to the placebo response. So this is a study that was published in 2001. So using PET and a radiotracer called raclopride, which binds to D2 receptors, you can measure dopamine release by scanning people before and after a challenge pharmacologically or behaviorally. So people with Parkinson's disease were given placebo or apomorphine, which is dopamine agonist. And after placebo, they had the same amount of displacement as they had with apomorphine. So we're seeing a real biological change. So the placebo response is biologically real in Parkinson's, and this might explain some of our findings, our results. We're still blinded, but we're seeing really a massive response. If you think back to the response rates, half of those hopefully have received placebo. So there's clearly some kind of placebo response going on. So then I kind of wanted to end, but I've got loads of time, by telling you about some patient quotes. So we just added a qualitative interview to really capture what's going on in terms of expectancy and how people coped with the ketamine. I could also mention actually that it was very well tolerated, which we didn't know whether it would be in Parkinson's disease. No serious adverse events that are related to the study, and just kind of, the expected side effects that we get from all our ketamine studies. So this is, some patient quotes. We've only done 4 interviews so far, but that it has really impacted their lives. I've unshackled myself. I can perform in the gym, pick up things better, partly because I don't have the emotional burden I carried before. With Parkinson's, you lose so much control. This gave me the ability to take control back with the way I deal with things. Okay, so then non-specific effects. So I think, especially for this population, taking part in a clinical trial has been quite empowering. They feel like they're helping, and it's given them confidence that they can do something. So that's a non-specific effect that we need to be aware of. Sometimes even just having somewhere to go, something to do. This patient, found that it was just nice to have structure, and I think that can be a thing with clinical trials in general, especially with depression, but maybe more so in people with Parkinson's disease. Then this patient, I did feel relief from my depressive symptoms, but I don't know if it was just from being heard, being understood. This was the first time that I had people who got it, whether I got placebo or not, whatever happened, it was effective for me. So I think this, again, with this population, patients have their neurology appointments and they're like 15 minutes. And most of it, I think, is around medication management and their Parkinson's symptoms, and non-motor symptoms and mood symptoms are generally not talked about. And I don't think they have an opportunity to tell them what it's like, actually to tell clinicians what it's like to live with this disease. So I think that is going to be one of the non-specific effects. And then, one patient just said that she just felt taken care of, which is great. And we had decided at the beginning of the trial that we would treat everyone nicely with empathy, and that's one way of doing things, but we're going to get more of these effects. But I think these effects, like Jerry was saying yesterday and others are saying, they're clearly there, and I think we can harness them, and that we can synergistically look at the specific effects, like the mechanistic work I presented at the beginning of the presentation. There's something specific going on, I believe that, but definitely there's non-specific effects, and especially with this population, I think we're going to see a huge synergy with both of those things that I think we can harness rather than try and diminish. I don't think that's going to be helpful for people. So there's like 4 minutes left, so loads of time for questions. So yeah, thank you. I just cut myself.
Fantastic. I'm gonna take the opportunity to ask the first question, if that's okay.
Yeah.
So I treat people with depression and lots of anhedonia, and I use quite a lot of Parkinsonian drugs to treat them. But if you look at the data, and I guess my experience is that the different drugs differ in how useful they can be in depression, right? So L-DOPA work very well, but the D2 agonists have quite a good evidence base for treating depression, and does kind of target, to some degree, right, the base pathology in this group. So are you controlling for that in your randomization? Or are you going to look at that? Because that's a...
We'll look at it. Mostly people are on carbidopa, levodopa, so not agonists. But yeah, we will control for that. But I think all but one have been on Parkinson's medication. Maybe 2, actually. The CEO, I think, was not on medication.
I think maybe 1 or 2, but remarkably, like pramipexole, I don't think we have anybody. The neurologists in our town are just not.
It's the same in the UK, but I don't know. In my practice, anhedonic patients works fantastically.
Same with me. I use pramipexole all the time, but none of these patients and neurologists don't use pramipexole. Yeah. I don't think any of our patients.
It's all carbidopa, levodopa, but different.
Pretty much everybody is on carbidopa, levodopa.
Yeah.
But they do, there's been studies, and anecdotally they say if they miss doses, then their depression, anxiety gets worse, and that could be because of they're worried about freezing, or their tremors get worse.
Yeah.
But I think there's something going on with dopamine, but it doesn't explain depression, I don't think, fully.
I like your argument that you should be nasty to your patients during the trials, because otherwise you get a massive placebo response.
Well, we decided to be really nice, and hopefully we can harness that. But whether we'll see a difference, I don't know.
There's one Rupert, and then there's somebody there as well.
Yeah, I was just interested in what you were saying about raclopride and that sort of justification for the placebo response in Parkinson's, when, of course, the defining feature is that they're going to have fallout in the basal ganglia.
Yeah.
So this, you're saying this persists despite that?
Well, yeah. So there've been so many studies showing that the placebo response is really big in Parkinson's. You'd think that if it's dopamine related, maybe it'd be lower because they've got less levels of dopamine. But it's thought that maybe damage to the dopamine system, it's unstable. I don't know exactly. But Jerry, what's your hypothesis?
Well, the hypothesis has been that there's probably, we're talking about prediction, how prediction plays into expectations, and the hypothesis is that patients have a disruption in their prediction. So they may be more sensitive to expectation changes. There's high rates of placebo response in Parkinson's studies.
Could the depletions in dopamine be related? Could the depletions in dopamine be related to an increased turnover in the terminals, and therefore you're getting a little bit more release in general? So that puts in a little bit of yeah.
This may have been addressed, but just to piggyback on the comments about pramipexole, if I recall, the first studies on antidepressant effect of those were in patients with Parkinsonism. Do you see, or did those studies show comparable rates of placebo response, or is there something about the idea of what it means to have a ketamine experience that?
Yeah. So sorry, what's the question?
The question is just with other antidepressant trials in Parkinsonian patients, do you see the same? And I apologize if I missed this.
Yeah. So antidepressants in general, there's not efficacy over placebo, and whether that's because they're not working or the placebo response is so high, we don't know. there is evidence of neurodegeneration in RAF-A nuclei, so I think serotonin could be involved, but I don't think that's explaining all of depression.
Presumably, you're excluding people with cognitive impairment.
Yeah.
Great.
Yes.
All right. One more question, maybe?
18. We did actually lower the MoCA, but that's because they didn't have like clear dementia or cognitive impairment, and they still could have informed consent, but they were just getting like 18 or 19 on the MoCA, so we lowered it from 21 to 18.
Okay, there's time for one more question over there.
Pardon my voice. This is a little bit of a self-interested question, but a curiosity question nonetheless. How many of your participants also have concurrent chronic pain and opioid use?
The things in the behavior scale, which is specific to Parkinson's. And we have seen in 3 patients, and they've told us that they have significantly reduced pain on taking off. But we haven't, we're also blinded as well, so we haven't fully looked at it.
I only ask is I'm wondering if you have enough of those patients to do a sub-analysis like what I had presented on the first day. That would be interesting to see with regards to placebo effect.
Mm-hmm.
Okay, yeah. That makes sense. Thank you.
Thanks very much,
Thank
Sophie.
you.
Really
Thanks.
interesting talk. So next up.